Today we are going to discuss the chapter Molecular Basis of Inheritance. Previously, we have discussed about the principles of inheritance, where we studied about the inheritance patterns and the genetic basis of inheritance. Today we are going to discuss the character, the chemical nature of the molecule responsible for inheritance. The topics that we are going to discuss are DNA structure, packaging of DNA helix, Griffith experiment, Hershey and Chase experiment. Now let us first see how the DNA was discovered. Initially, it was identified by Friedrich Mister in 1869 and he called it nuclein. In 1953, Watson Crick he discovered the double helical structure of DNA with the help of the technique which is called X-ray diffraction data, which was developed by Wilkins Franklin. Before we go into the detail of DNA structure, let us see where the DNA is located. The DNA is located in the cell. The cell contains the nucleus. The nucleus contains chromatin material. The chromatin material, during the cell division, it organized into chromosomes. The chromosomes contains the DNA. Now let's recall the chemical nature of DNA. As you can see in the diagram, there are three important components: the nitrogenous bases, pentose sugar, and phosphate group. The nitrogenous base is attached to sugar with the help of a linkage which is called glycosidic linkage. The combination of nitrogen base to sugar forms nucleoside. The phosphate group is attached to the 5' OH of nucleoside with the help of a linkage called phosphodiester linkage. Now let us see that how the two mononucleotides are joined together to form dinucleotide. 3' 5' phosphodiester linkage makes the two nucleotides join together. There is a 5' free phosphate group and the backbone is made up of sugar and phosphate. Let us understand with the help of the following picture. As in the picture, you can clearly see the nitrogenous base is attached to phosphate sugar. The phosphate sugar is attached to the phosphate group, and there is a linkage between the 3' and the 5' between the two nucleotides. Now let's have a look on how the polynucleotide looks like. Now this is a very simple model which you can also make, which shows the clear anti-parallel character of DNA. The 5' to 3' and from 3' to 5'. Let us see how the DNA molecule looks like. It's a long polymer of deoxyribose nucleotides. Now let us see the salient features of DNA. The two polynucleotide chains, they are anti-parallel. The backbone is made up of phosphate and sugar. The anti-parallel polarity, that is, it is from 5' to 3' and from 3' to 5'. The nitrogenous bases are joined together by hydrogen bonds. That is, the A pairs with the T and G pairs with C. There is a difference in the hydrogen bonds between AT and GC. As you can clearly see in the picture, between A and T there is a double bond and between G and C there is a triple bond. So the following diagram shows the antiparallel features of the DNA, 5' to 3' and from 3' to 5'. Now let us study about the coiling of DNA. The coiling is done in the right-handed fashion, as you can see in the picture. The pitch of the helix is 3.4 nanometer. And in each turn there are 10 base pairs. So the distance between each base pair becomes 0.34 nanometer. Another very important feature is that how the stability is confirmed on the DNA. It is because of the plane of one base pair stretched over the other, so that the stability is confirmed. Another important feature Let us study what is the importance of base pairing. Number one, it provides complementarity to each other. And second, it helps the sequence of other strands that can be predicted. That means you can predict the base pairs of the other strand if you know the base pairs, the base nitrogenous bases in the previous strand. As we know that C always pairs with G and A always pairs with T. So if a one strand contains A, G, C, then the other pairs, because of the base pairing, will be able to predict the nitrogenous bases of the previous strand. Now let us see some interesting facts about the DNA. In the bacteriophage, like 5X174, it has 5,386 nucleotides. And in E. coli, the DNA base pairs are 4.6 into 10 raised to the power 6. On the other hand, in human DNA, the base pairs are 3.3 into 10 raised to the power 9 base pairs. And the length of the DNA is 2.2 meters, whereas the size of the nucleus is only 10 raised to the power minus 6 meters. Now you can imagine that how the DNA is packaged in such a small nucleus. The size of the DNA is 2.2 meters. It has to be packed into such a small nucleus. Let us study further. So we now deal with the topic that how DNA is packed in a cell. Here we'll study separately the prokaryotes and the eukaryotes. In prokaryotes, the DNA is held with proteins. As you can see in the picture, the DNA is organized into a region called nucleoid. There is no true nucleus. On the other hand, in eukaryotes, the DNA is present in a well-defined nucleus. Now, if we go little detail into prokaryotic DNA packing, we see that the DNA is packed in the form of loops. So there is so much of coiling. On the other hand, in eukaryotes, the DNA helix consists nearly 200 base pairs. And here we see the repeating unit of chromatin. So on this picture, it looks like as beads on string on the chromatin, as in the form of a necklace. Now let us study the structure of chromatin. As we know, it is found in the nucleus. It looks like thread-colored bodies, and these chromatin only forms the chromosomes during cell division. You can see clearly in the picture the chromosome and the condensed chromosome structure, where the chromatin materials, which is thread-like during the prophase stage, how much it is condensed during the metaphase, which we see under the microscope during the metaphase stage as chromosomes. Now it has been found the DNA is packed in the cell in the form of nucleosomes. As you can see in the picture, the nucleosomes contains 8 protein molecules, and that forms an octamer. Now let us go detail into the structure of nucleosome. Now what is an octamer? This is positively charged basic proteins called histones. Octamer is made up of 8 histone protein molecules. These histone protein molecules are positively charged basic proteins. The DNA is wrapped around the histone octamer. These 8 protein molecules are H2A, H2B, H3 and H4, which are present in 2 copies. Now between the 2 nucleosomes there is a linker DNA, and that is how we get a bead on string structure. Now there are 2 different regions in the chromosomes, the euchromatin and the heterochromatin. Let us see what are the differences between the euchromatin and heterochromatin. In euchromatin, the chromatin material is loosely packed. It is transcriptionally active and it stains light. On the other hand, heterochromatin is the region which is tightly packed, inactive, and it is darkly stained. Now let us study the euchromatin and heterochromatin with the help of the following figure. The orange colored regions that indicate the heterochromatin, the centromere that joins the two arms of the chromosomes, and the blue region that you see is the euchromatin. So now we have studied the detailed structure of DNA and how the DNA is organized into the chromosomes which we see during the cell division. Now a very important question is that what is the chemical nature of the DNA? We have studied the chemical nature, but how it was proved that DNA is the genetic material. So now let us study certain experiment that has led to the proof that DNA is the genetic material, not other macromolecules like RNA or other macromolecule like carbohydrate. So now let us study the search for genetic material. The first experiment that was performed for the search of genetic material was by GRIFFTH. In 1928, he worked on the bacteria Streptococcus pneumonia that causes pneumonia in rats. Now, while he was experimenting with these bacteria, he found a surprising change in the bacteria. These bacteria, Streptococcus pneumonia, it occurs in two forms. The one is the S strain, which is called the smooth strain, and the other one is the R strain, which we call the rough strain. The S strain is smooth because it has a polysaccharide capsule on the bacteria. On the other hand, in the case of rough bacteria, they lack this wall and therefore they appear rough. These smooth bacteria, they are virulent. That means they causes the disease. On the other hand, the R type is non-virulent. Now let us see how he performed the experiment. In the step 1, he injected the S strain into the mice. Now since the S strain was virulent, so the obvious result was the mice died. In the step 2, he injected the R strain, the living R cells. But since it was non-virulent, the mice lived. In step 3, he injected heat-killed S strain. And what he found? He found the mice live. In the step 4, he mixed the S strain heat-killed with the R strain lived. And to his surprising, he found the mice died. How it happened? So he concluded that the heat-killed S strain bacteria changed R strain bacteria into S strain. And there is some transforming principle that has been transferred from heat-killed S strain. That means the S strain was capable to transform the R strain into S strain, and that is how the mice died. But the biochemical nature was not defined by GRIFFTH. So in the next experiment, the characterization of transforming principle was performed by 3 scientists, Avery, MacLeod and McCarthy, 1933 to 44. They purified proteins, DNA, RNA from heat-killed S cells. Now to their surprise, they found it is only the DNAs which inhibit the transformation. On the other hand, proteases and RNases did not affect the transformation. So that clearly proves that DNA is the genetic material. So their observation were that S bacteria changed live R bacteria to S bacteria, and so the mice died. And that was characterization by the scientists that the DNA is the genetic material. But the conclusive proof only came by the experiment performed by Alfred Hershey and Martha Chase in 1952. They worked on bacteriophages and E. coli. Now let us see how they performed the experiment. The first step of the experiment was infection. That is, the bacteria were infected with the bacteriophage. Now they use two types of radioactivity. The one was P32 and the other was S35. Now they use two types of radioactivity. The one was P32 and the other was S35. After infection, the next step was blending and centrifuging. To their surprise, they found, after centrifugation, the two components, the one was supernatant and the other was the pellet. They found when they used P32, they found the radioactivity in the pellet. Now what does this pellet contain? The pellet contained the bacteria. On the other hand, they found S35 radioactivity in the supernatant. So the following diagram clearly shows the two experiments were performed parallel, in which they used P32 and the other one they used S35. The supernatant shows S35. On the other hand, the pellet shows P32. Now a very important question here, that why they use only phosphorus and sulfur? Can you guess? Yes. The DNA contains phosphorus, therefore they used radioactive phosphorus, and the protein contains sulfur, so therefore they use sulfur. Now since the supernatant contains the protein and the pellet contains the phosphorus, so what is the clear indication? Now let us see the observation of the experiment. The bacteria infected with P32 were found radioactive, so the DNA was passed from virus to bacteria, and bacteria which were infected with S35 not radioactive, so that clearly shows that protein is not passed. So now let us see the summary. DNA is long polymer of deoxyribonucleotide. The three components of DNA are nitrogenous base, pentose sugar, phosphate. In RNA, uracil replaces thymine. The DNA is stable due to hydrogen bonds. In eukaryotes, DNA packed into nucleosomes, transforming principle proved by Griffith experiment, and conclusive proof of DNA as genetic material by Harshay and Chase. Now you can check your understanding by the following. DNA is present in eukaryotes. The two options, the cytoplasm or the nucleus. The phosphate group is linked to nucleoside through bond. DNA double helical structure discovered by. Differentiate between euchromatin and heterochromatin. The packaging structure of DNA in eukaryote is called. Explain Harshay and Chase experiment. And the last question, that why P32 radioactivity was found in pellet. Thank you, students. Now